Hello and welcome to this video on machine sensors versus those of a human. On the side of our future robotic overlords is the analytical lab machinery, comprised of high liquid pressure chromatography, gas chromatography, mass and isotope specific spectroscopy. This channel welcomes our future robotic overlords and wishes them well in the coming match. The machinery making up this team ranges from 100,000 euros and more for each unit and a dedicated lab. On the other side of the field is the home team, the squishy meatbags known as humanity. The team is comprised of a head with a nose for smelling, mouths that taste, and a sense of touch. None of these are worth much individually or as a transplant tissue. Now, to be slightly more serious, the human sense of taste and smell is something amazing, especially so when compared with the mental capacity of chimpanzees typing randomly. It can differentiate between the smallest amounts of a wide range of compounds, and this is in a very compact form. They require no prior learning or calibrating. The ability to differentiate between flavors and identify each is a combination of the 5 senses. After taste is combined with smell, flavor is produced. If the sense of smell is impaired, taste is dulled. By comparison, lab equipment such as a chromatograph and isotope machine require considerable room and are incredibly large. These machines need known samples or a control comparison, along with regular calibration. From this it should be apparent that the large expensive machines suffer from shortcomings by comparison, but are still used as an unbiased control mechanism for measuring of unknown and known quantities. The first human sensory function is taste. It is well established that the human taste covers 5 flavors: sour, salty, sweet, bitter, and savory, the last of which is also known as umami or MSG. All of this separation is done in the space the size of a small fruit. Not only separation, but identification in a volume the size of a small melon. Substances are freed in the mouth and come into contact with nerve cells. It activates the nerve cells, and these change specific proteins in the wall of the sensory cell. This change causes the sensory cell to transmit messages about the substance. These in turn activate further nerve cells, and these nerve cells then pass information on, and this forms a particular perception of flavor in the brain through the pattern of activation. There are 3 forms of nerve cells: fungiform papillae, circumvallate papillae, and foliate papillae. About half of the sensory nerve cells react to several of the 5 basic tastes, and these only differ by having a varying level of sensitivity to the different basic tastes. The other half of the sensory nerve cells and corresponding nerve fibers are specialized, and these react to only one taste, and these provide the differentiation in the intensity of the stimulus. Molecules released by substances stimulate these receptors, there being more flavors in the environment than there are receptors, and it is any given molecule that can stimulate a combination of receptors, creating a unique representation in the brain, this representation and pattern becoming a flavor. This is also important when it comes to the sense of smell. The small molecules that are released during oral consumption also activate parts of the olfactory sensory system, and this is done through 2 pathways. The first is through the nostrils, and the other pathway being the previous oral system, which creates aerosolized molecules that travel up the olfactory cavity, this being the second human sense, smell. It is well established that the human sense of smell is extensive. It can isolate, detect, and identify trace amounts of gases and vaporize small solids. The ability to smell comes from specialized sensory cells called olfactory sensory neurons, and these are found in a small patch of tissue at the back of the nose. Olfactory receptor cells are neurons that have knob-shaped tips, and these are called dendrites. Olfactory hairs that bind with odorants, being the aerosolized small molecules, cover these dendrites. When an odorant stimulates a receptor, the cell sends an electrical impulse to the olfactory bulb, and then this goes on to the brain. The 2004 Nobel Prize winners were able to prove that over 1,000 genes, or roughly 3% of the human total, coded for olfactory receptor types. Their work was able to show that every olfactory receptor cell has only one type of receptor on its surface, and that each receptor type can only detect a small number of related molecules, and then respond to some with greater intensity than others. It is even believed that the ability for humans to smell can identify when women are ovulating. You'll find a link to that below. This summarizes the two primary human senses that relate to analytical lab equipment and the machine senses. The first of the lab-based equipment is the HPLC-MS, or high-pressure liquid chromatography mass spectroscopy. Compare the two senses discussed so far with the following equipment. It is a machine designed to use a method called high-pressure liquid chromatography. The model seen here has a mass spectrometer fitted. Mass spectroscopy will be discussed later. HPLC works on the principle of electron negativity and positivity. This is done by dissolving the mixture or compounds in a carrier fluid that has a certain charge to it. The liquid moves through a cylinder of a generally known charge that the liquid interacts with. This either slows down the movement or speeds it up. It is very much like distillation, except it can increase or decrease the speed of movement. The product then gets to a point in the cylinder where the compounds are too slow in comparison to one another, and so separate into what is known as a fraction. These fractions eventually make it to the end of the cylinder, where it is either dumped as waste, put into a detection device, or placed into storage vials as a more pure isolate. The next stage after measurement can be the use of other detectors or a mass spectrometer. The machine can generate a timeline of when each product comes off the cylinder, and if a known compound is used as a control, it can then be measured against these results to produce a comparable plot. Some machines, and most newer models, can be programmed with a known standard curve and extrapolate how much is in each point based on the intensity of feedback, much like how the human sense of taste and smell work. The next piece of lab-based equipment is the GCMS, or gas chromatograph mass spectroscopy. This is much like HPLC, but it uses a gas instead of liquid. This is good for volatile compounds. It also recreates much of the functionality of the human sense of smell. Gas chromatography is like liquid chromatography. These are both far more complicated and require their own dedicated video to explain in appropriate detail. However, gas chromatography has certain advantages in that it can work on both toxic and minuscule amounts of gas or other highly volatile compounds that might be toxic to humans. The final piece of lab equipment is the mass spectroscoper. Mass spectroscopy charges ions and bombards a sensor with them. It detects the weight of the molecule and compares this with the charge. This means a purified sample can have the number of molecules, their charge, and possibly their nature and interaction identified through the laws of probability, an extrapolation on the nature of the number of charges in each particular thing. Arguably, the human body has no response to this or a comparable measurement. However, it does have certain traits that are arguably in contrast, and these are the ability to identify the differences in compounds that are closely related that the machine is unable to compete with. This is particularly obvious in chirality and cis v trans molecules, such as limonene. This is the flavor in lemon, or what is directly responsible for the flavor in lemon, and orange. However, these two are very different flavors, and it is due entirely to the molecule's rotation, which is unable to be identified through machinery, but the human sense of taste can differentiate between the two, despite the fact that the two are effectively the same molecule as far as the composition of elements goes. The final piece of lab-based equipment are isotope machines. Isotope ratio mass spectroscopy is a special subfield of mass spectroscopy. Mass spectroscopy methods are used to measure the abundance of certain isotopes. Isotopes are the differing level of charge in the same element on the periodic table based on the amount of energy that was input into the molecule during development of the Earth's crust. Between the human senses described and the lab-based machine, there are obviously more possible options, and these could be discussed, but the two human senses and the lab-based machinery discussed so far cover the primary senses and means of measuring things within a lab. From what has been said so far, the biggest drawback for machinery is size and cost by comparison to the human sense. Once these two problems are addressed, it will become advantageous to use machinery in all aspects of research for the reliability, consistency, and quantification of everything. By contrast, the human sense of taste and smell are limited in comparison to other animals. The mitigating factor remains the interlinking and adaptation from several disparate organs. This allows the human sense of taste and smell to identify fractional amounts of compounds in wine and other beverages, where large-scale machinery with finely adjusted procedures and experiments are needed to identify. It is this particular ability of the human sense of taste and smell to identify a very specific, small compound without having to specifically isolate it within the larger solution of flavors that makes the human sense of taste and smell special. As with any machine, these particular detection devices cannot work beyond their scope. In this case, small molecules can hide behind large ones, and the machine can only go so low in concentration. The human senses can often detect some of the lowest concentration molecules, where a machine cannot. For example, flavor compounds in wine, such as tannins, or variations of tannins, may have a significant impact on the final result, where a machine may not be able to identify their presence at all due to such low and small amounts. The other side of this equation is the human sense cannot quantify the amount or detect it consistently. As a result, in the near future, our robotic overlords will arrive and all will be right with the world.
Thank you for watching this video. Please post any comments, questions, or suggestions you may have below.